Much for the introduction. I appreciate it. That's great. All right, let's see if I can share my screen again successfully there. All right, you guys see what you're supposed to see? Fabulous. All right, thank you. So one other announcement is that I am moving next summer from Florida State to Texas A&M in College Station. So if anybody wants to do nitrogen and phosphorus cycling, come into Texas. So what I want to start by talking about is the overarching research question in my lab is, what are the sources of nutrients fueling carbon fixation in the oligotrophic ocean? So the parts that we call sort of ocean deserts or regions where inorganic forms of nutrients are scarce. So one way to think about this, let's see, I'm going to move there. Oh, can I move you guys off? No. All right. Well, so you get to see yourselves. That's okay. Okay. So there's this picture, of one way to sort of image the biological pump, right, is using satellites. And so this is a false color map of surface ocean chlorophyll. There's also chlorophyll on land. But one of the things that you can see if you just look at the ocean is that chlorophyll concentrations are not equally distributed around the ocean, right? We see higher abundances of chlorophyll in the high latitudes and also in the equatorial Pacific and also in coastal systems, all right? And to first order, this looks a lot like the surface ocean distribution of nitrate concentrations. So this is a map from World Ocean Atlas nutrient data of surface ocean nitrate concentrations ranging from 0, the blue color, in most of the low and mid latitude regions, to up excess of 25 micromolar in the Southern Ocean. But there's a lot, to first order, these two maps of surface ocean chlorophyll and surface ocean nitrate look very similar. So what's the question, right? Well, these areas that have really low concentrations of surface ocean nitrate, and I could show you a map of phosphate and it would look very similar. These regions we call oligotrophic gyres. So they're the center of the tropical and subtropical circulation systems. And what's interesting is they still support really high rates of carbon fixation and export production. And so Steve Emerson compiled this table almost 10 years ago, where he took a bunch of metrics of largely geochemical metrics of annual net community production. So determined by oxygen, carbon, isotopic composition, nitrate drawdown, and a bunch of metrics. And he compared regions like Ocean Station Papa, which is sort of up here in the Northeast Pacific, the hot site, so near Hawaii, equatorial Pacific in the east and west ends of the equatorial Pacific, the subtropical, subarctic, North Atlantic, the Bermuda site, and coastal California. And what's interesting is that the rates, I don't know if you guys, there we go. Okay, so if you take the average of these, what you see is that they are largely similar. And so this was really, a lot of people have known this for a long time, but I love this table because I think it really underscores just how high rates of productivity are in these oligotrophic waters. And so the overarching research question in my lab is, what supports this carbon fixation, right? How do you actually have phytoplankton meeting their nutrient needs when inorganic forms of nutrients are scarce? And so my research focuses on two main sources of nutrients. And so one thing that I do a lot of is what we call delta N15 budgets. And so I'm going to introduce you to those. And this is a way to quantify the relative importance of subsurface nitrate versus nitrogen fixation as nutrient sources fueling carbon fixation and export production in oligotrophic gyres. Another theme of my research is trying to understand and quantify the role of dissolved organic nutrients supporting carbon fixation in these gyres. So the idea here is that the net production of dissolved organic nitrogen and dissolved organic phosphorus largely happens in these nutrient-replete upwelling systems. So in the Southern Ocean, in coastal systems, in the equatorial Pacific. And then these dissolved organic nutrients are advected into the center of the gyres. And so I'm developing tools to try and quantify how important organic nutrients are as a nutrient source for fueling carbon fixation in those oligotrophic gyres. So what I want to do is start by introducing you to delta N15 budgets if you haven't met them already. Okay. And so I love delta N15 budgets because they tell us two really important pieces of information. The first really important piece of information that they tell us is the relative importance of subsurface nitrate and N2 fixation for supporting export production in oligotrophic systems. So this is my cartoon schematic of the euphotic zone, and then this is my version of a phytoplankton that sinks out of the euphotic zone. Okay. The second really important thing that a delta N15 budget can tell us is a strictly geochemically based rate of N2 fixation. Okay. And so what I like to do is do these delta N15 budgets at sea or on cruises or research expeditions where I have colleagues like Rachel and other people who are doing bottle-based rates, so incubation-based rates of N2 fixation, as well as using molecular tools to try and characterize the importance of diazotrophs or nitrogen fixers in oligotrophic systems. And then I like to compare and try and understand if we're getting the same story from all three tools, which are completely independent of each other. They don't know anything. They're not influenced by the results of each other, and see what we learn. Okay, so this is really strictly a geochemical approach. All right, so the trick about delta N15 budgets is we have to make a few assumptions. Okay, so what we're basically doing is a 2N member mixing model. So if you've never done one of those, it's like calculating your GPA, right? Maybe you guys don't have to do that in Sweden, but Americans are very focused on their grade point average, right? So at any rate, the point is that we assume that there are two quantitatively important sources of new nitrogen fueling export production, and those are subsurface nitrate and N2 fixation. Okay, and then what we want to do is take advantage of the fact that those two sources of nitrogen have distinct nitrogen isotopic compositions and compare them with the delta N15 of the sinking flux. And so all we're doing is comparing the isotopic composition of sources of nitrogen in with what leaves. So what goes up or comes in has to be equal to what leaves the system, right? So we have to make a few assumptions. One is that these are the only two important sources of nitrogen fueling phytoplankton growth and export production. The second is that export production is well represented by the material that we capture in a floating sediment trap. And there are some problems with that I'm happy to talk about. But to first order, it seems fair and is a reasonable assumption. The next assumption that we have to make is that the system is in steady state on the timescales that we're out there evaluating this over. And that's the best that we can do. So that is to say that fluxes in have to balance fluxes out during our time on station when we have sediment traps deployed. Okay. And so what I'm going to do is go through an example delta N15 budget, and then I'm going to show you results from the ocean from a cruise that actually I was on with Professor Foster, and then talk to you about results from other delta N15 budgets from around the world. Okay. And so when we do a delta N15 budget, we assume that we know the delta N15 of newly fixed nitrogen, of nitrogen fixer inputs based on a lot of culture work that people have done. And so we assign the delta N15 of newly fixed nitrogen to be -2 to 0 per mil. For the sake of this example, I'm going to call it 0 per mil. What I have to do, the delta N15 of subsurface nitrate varies around the globe. So when we go to sea, we have to collect water column profiles, and then we bring samples back home to the lab. And in my lab, we measure the delta N15 of subsurface nitrate. We also have to deploy floating sediment traps to capture the sinking particulate nitrogen and see what its delta N15 is and compare it with the two sources of nitrogen. So say we go somewhere in the ocean and we measure the delta N15 of subsurface nitrate and find it's 8 per mil, and we know that the delta N15 of newly fixed nitrogen is 0. What we want to do is compare that with the delta N15 of the export flux, the sinking particulate nitrogen. Okay, so let's say we go somewhere and we find that the delta N15 of the sinking particulate nitrogen flux is 4 per mil. What that would tell us is that half of the carbon fixation and export in the system was supported by subsurface nitrate and half was supported by N2 fixation. Because if you mix something that's 0 and something that's 8 in equal proportion, you're going to make something that's 4, right? So here in this example, N2 fixation would support half of export production. Let's say we went somewhere else and we measured a delta N15 of the sinking flux and it was 2 per mil. That would tell us that 75% of export was supported by N2 fixation and 25% by subsurface nitrate. Okay, so that's how we get the first really important piece of information from a delta N15 budget is the relative importance of subsurface nitrate and N2 fixation for supporting export. The next really important piece of information that we get is a geochemically based N2 fixation rate, okay? So let's say we go somewhere and all we're gonna do is take the magnitude of the sinking particulate nitrogen flux. So how much sinking particulate nitrogen is captured in our sediment trap. So let's say we were somewhere and we measured a sinking flux of 400 micromoles of nitrogen per meter squared per day. All we do is multiply that by the fractional importance of N2 fixation, right? Because our delta N15 budget is telling us how important N2 fixation is for supporting that export, and this is the magnitude of that export. So when we multiply those, we get a strictly geochemically based N2 fixation rate. So in this system, we would estimate that nitrogen fixation rates were 300 micromoles of nitrogen per meter squared per day, okay? So this is something that we've done in a bunch of places. Here is an example from, this is actual data from the Eastern Tropical South Pacific. And so, like I said, Rachel was on this cruise with me when we collected these samples. So the y-axis is depth from 0 to 400 meters. The bottom x-axis is nitrate concentration from 0 to 35 micromolar. And nitrate concentration is in the open circle. So you can see it's pretty oligotrophic. There was basically no nitrate in the euphotic zone. The upper x-axis is delta N15 from -2 to 20 per mil. The delta N15 of subsurface nitrate is here, and it's about 10 per mil throughout the water column. I'm also showing you this is the delta N15 of the sediment trap material. So it was about 8 per mil. And up here on the top x-axis, this arrow represents the delta N15 of newly fixed nitrogen. So here we actually assign it a delta N15 of -1. So what you can tell just qualitatively is that the delta N15 of the sediment trap material is much closer to that of subsurface nitrate than it is to N2 fixation, right? And so if you do the calculations, what you find is that at this station, nitrogen fixation supported about 20% of export production, and subsurface nitrate supported about 80%. But the thing is, this station was on the edge of the ultra oligotrophic Eastern Tropical South Pacific gyre, where there's super low rates of photosynthesis. And so the magnitude of export production at this location is very low. And so what this corresponds to is an exceedingly low nitrogen fixation rate of about 10 micromoles of nitrogen per meter squared per day. Okay, so what we found in the Eastern Tropical South Pacific gyre is very low rates of N2 fixation. This is similar to results we had at other stations and over the course of 2 years, okay? But then what I told you a little bit ago is that we like to compare the results of the geochemical estimates of nitrogen fixation with bottle base rates or other tools and techniques to try and get a sense of how much confidence we should have in our results, right? And so what I'm showing you here are results from the Eastern Tropical South Pacific from 3 stations we occupied in 2010 and 3 in 2011. In the blue are the geochemical based and delta N15 budget based results. So F is the fraction of export production supported by nitrogen fixation. And you can see in 2010, we found no role for nitrogen fixation supporting carbon fixation or export production based on the delta N15 budget. And in 2011, at 2 stations, we did find a small role. These are rates of nitrogen fixation from the delta N15 budget, and these are very low. If you compare these with the 15 N2 uptake, so sort of the traditional bottle-based rates of N2 fixation, what's interesting is that these are still pretty low if you put them in the global context of all the rates that we've seen, but they're always higher than the delta N15 budget-based rates. And that's something that we have seen a lot of, is that the bottle-based rates are always higher than the geochemically based rates. But what we wanted to do next was compare these with molecular data. And so this is work that was done by Kendra Turk-Kubo, a colleague of ours at the University of California in Santa Cruz. And what she did was she characterized the diazotroph community composition using molecular tools. And what she found is that the typical cyanobacterial diazotrophs that we know and love, like Trichodesmium or Crocosphaera or the eucins, were not there. They were just not present in the Eastern tropical South Pacific. She did find some low abundance of heterotrophic diazotrophs. But what she did next was really interesting. And what she did was she took the estimated abundance of the diazotrophs based on her molecular data, how many gene copies she found. And then she went to the literature and she took estimates from the literature for the diazotrophs that she did find and their per cell rates of nitrogen fixation. So she multiplied the abundance of the diazotrophs by their per cell rates of nitrogen fixation. And what she found is that the estimated rates of nitrogen fixation from that approach were far lower than the bottle-based rates. And so based on the diazotrophs that she could find, using her primers and her tools and techniques, she couldn't account for the bottle-based rates. So instead, the molecular data was a lot more consistent with the Delta N15 budget data. Okay. This is an example from the Eastern tropical South Pacific, but I've done these kind of Delta N15 budgets in a lot of places, and so have other people as well. And one of the conclusions that I have come to from doing these kind of measurements for a long time is that subsurface nitrate always seems to be supporting more export production and more phytoplankton growth than we have traditionally accounted for. And it's a little tricky because we don't understand the mechanisms by which subsurface nitrate is making it up to the euphotic zone. There's some evidence that there's vertically migrating phytoplankton. There's also, I think, a lot of evidence for physical mixing of nitrate up from the subsurface into the euphotic zone. But these are things that we're still trying to resolve. The only place where there's an exception to this, where N2 fixation supports the majority of carbon fixation and export production, is in the Western tropical South Pacific. So just to quickly summarize, Delta N15 budgets have been done by myself and others at Bermuda, at Hawaii, in the Eastern tropical South Pacific, in the Eastern North Atlantic, in the Gulf of Mexico, in the Gulf of California. And in all these cases, nitrate is supporting the overwhelming majority of export production in these locations, okay? And so again, I think there's a lot of really compelling evidence that there's more subsurface nitrate making it up to surface waters than we have traditionally accounted for in these oligotrophic gyres. When nitrogen fixation is important, and usually nitrogen fixation has been detected in these budgets, it's most important in the stratified summertime, but it usually is supporting something like 25% of export and it's usually focused in the summer, okay? And again, the exception to this is some work that I've been doing with a French colleague, Sophie Bonnet, and we have been finding really high rates of nitrogen fixation in the Western tropical South Pacific. So some of the highest rates in the global ocean. And so this is a project that I did with Sophie, and I've just got the results of the Delta N15 budget summarized up at the top, but the location here, it's about 20 degrees south between Noumea and Tahiti, and here's the coast of Australia for reference. So in this Western tropical South Pacific, but this spans a very wide range of longitude. And the results of our Delta N15 budget are here. So the fractional importance of N2 fixation for supporting export production, you can see at this station and at this station, half to the majority of exported carbon was supported by nitrogen fixation based on our Delta N15 budget results. Our estimated rates of nitrogen fixation are here. There was no N2 fixation detected at in the Delta N15 budget at this station, just to the east of Station B. And then these at the top are the bottle-based rates of nitrogen fixation. And so they are always higher than these, but to first order, the patterns that we're seeing are consistent. So very low rates over here and pretty significant rates at stations A and B. And so we had predicted that there would be higher rates of nitrogen fixation to the west of this transect than to the east because of atmospheric dust deposition. There's decent amounts of dust here, which nitrogen fixation has very high iron requirements for the enzyme that carries out nitrogen fixation. So we know that that is the sensitivity of this process. But what we were surprised about was how much nitrogen fixation was going on at this station B, especially compared to station C. And so my French colleagues went out again a few years later in a project that they're calling Tonga. And the results of this are coming out this year. And so one of the things that they wanted to understand was, what are the sources of iron supporting these really high rates of nitrogen fixation here? And when they were on the previous cruise in the Outpace project, they thought they detected signals of shallow hydrothermal vents supporting or supplying iron to the euphotic zone. And they didn't expect that when they went out on the earlier projects. So they weren't really equipped to definitively demonstrate that. So then they went out again to really try and collect all the data that they needed to support this. And so these are stations that they had also in the Western Tropical South Pacific. And so the results that we have now, I'm showing you profiles of water column nitrate, delta N15, and the delta N15 of sediment traps. So again, results of delta N15 budgets that I did with them. And here what you see at a couple stations that were located very close to some very shallow hydrothermal vents are very low delta N15 of the sinking flux. So around minus 0, they're actually negative. They're less than 0 at 2 different stations. And our subsurface nitrate is also very low in delta N15. But then the fractional importance of N2 fixation for supporting export is up here. So it's about 65% here, and about 90% of export was supported by N2 fixation, which this is the first time we've seen this anywhere, except for in the previous study when we saw really high rates of export production supported. So this is the only location in the world, I would argue, where we've seen results like this. So this has been really exciting. And Sophie published a paper in Science earlier this year that we contributed to. And what's nice is that I think the delta N15 budget results are consistent with the bottle-based rates as well as with the molecular abundance. So here is, I apologize that it's hard to see, but at some of these sites where we're over a shallow hydrothermal vent, they have really high chlorophyll abundance relative to the stations to the west and east. The suspended particulate nitrogen concentrations are high. Nitrogen fixation rates, these are in up to 30 nanomoles of nitrogen per liter per day. So very, very high rates of nitrogen fixation. Trichodesmium gene copies are upwards of 10 to the 6 NIF gene copies per liter for trichodesmium. So trichodesmium seem to be the dominant diazotroph here. And then this is dissolved iron concentrations. And these contours correspond to about more than 2 nanomolar, which is really high for these kind of waters. And this is at a depth of 150 meters. And so it seems that the iron from the shallow hydrothermal vents is reaching the euphotic zone and supporting really high abundances of trichodesmium that are contributing to these really high rates of nitrogen fixation and export production. The other thing that's really interesting to me is that the delta N15 of nitrate changes a lot between the Western and Eastern Tropical South Pacific. So this profile that I showed you a few minutes ago was collected at 20 degrees south and 100 degrees west, so at the eastern end of the Eastern Tropical South Pacific. And these samples are from about 165 east, and so at the opposite end of the basin. And the delta N15 of subsurface nitrate decreases from 10 per mil to less than 2 per mil, which this is some of the lowest nitrate delta N15 I've ever seen in the water column. So to me, this is painting a consistent story that there are really significant rates of nitrogen fixation that are contributing to a lot of export production, and they're significantly changing the geochemistry of the region. And so this is really exciting to me. So to summarize the results of the delta N15 budgets, in most parts of the global ocean, I would argue that subsurface nitrate is supporting the majority of export, even in stratified systems. The exception to this is the Western Tropical South Pacific, where we're finding really high rates of nitrogen fixation that support export production. But the second part that I want to very quickly talk to you about, and we had a couple papers published yesterday, so there's some new results hot off the presses and I'm really excited about, are the role of organic nutrients in supporting carbon fixation and export production. Because, you can have all this nitrogen fixation out in the Western tropical South Pacific, but diazotrophs, they don't just need iron, they also need phosphorus. And if you look at surface ocean phosphate concentrations in the Western tropical South Pacific, they're very low. But we've known for a really long time that phytoplankton in general, and diazotrophs in particular, are pretty adept at using dissolved organic phosphorus as an assimilative phosphorus source, okay? And so people have done a lot of really interesting studies at sea. But one thing that we've been lacking is sort of a broad scale picture of dissolved organic phosphorus concentrations in the global ocean. And that's largely because there aren't automated methods to measure DOP concentrations. And so one of the things that I've been working on, and I want to give credit to my PhD student, Zhou Liang, he's done a lot of the work that I'm going to be showing you next, and it's really fantastic work, is last year we published this global dissolved organic phosphorus concentration data set. So this is the first time there's been a high quality DOP concentration data set. We've made hundreds or thousands of DOP concentration measurements in my lab, and Joe has been leading this effort. And so this has given us the first sort of global scale picture of dissolved organic phosphorus concentrations in the ocean. And so this database is public. All this data is open access, and it's published in a couple different places, so everybody has access to it. But you can see where we have data in different depth sections of the global ocean. And using this newly published data set, what Zhou has been able to do is try and understand what controls surface ocean DOP concentrations in the global ocean. Because our hypothesis is that DOP is really important as a phosphorus source for phytoplankton in the oligotrophic gyres, right? And that's based on a lot of work that a lot of people have done for the past 20 years, right? But we haven't had the global scale data sets to really evaluate this. And so this is a paper that we published last year, and what we found, unsurprisingly, is that phosphate concentration is the dominant control on surface ocean DOP concentrations. So when there's a lot of phosphate around, DOP concentrations basically remain constant because phytoplankton would prefer to use phosphate than DOP, okay? But when phosphate concentrations drop, phytoplankton start consuming phosphate. But we also, what was unexpected, and Zhou gets all the credit for figuring this out, is that there's also a role for iron in this. And we think it's both because of the iron requirement for phytoplankton in general, but also there are some forms of the enzyme alkaline phosphatase that require iron. So alkaline phosphatase is an enzyme that allows phytoplankton to access some forms of dissolved organic phosphorus, okay? And so this is a figure. The x-axis is P star, which is a metric of phosphate stress or limitation, or we call it excess phosphorus. So when P star is high, there's a lot of phosphate in the water column, especially compared to Redfield stoichiometry and surface ocean nitrate concentrations. And then the y-axis is upper ocean DOP stock. So you can just think of this as DOP concentration. But the idea here is that when there's basically a lot of phosphate in surface waters, DOP can accumulate because phytoplankton don't need to use it. They have phosphate that they can use. But once phosphate is low, DOP stocks are low as well, okay? And what we have here are different ocean regions. And so the colors correspond to different ocean basins, with these regions being the Pacific and the blue being the Atlantic. Okay. And so the first primary control on surface ocean DOP concentration is phosphate concentration, okay? But we also found kind of a complicated role for iron, and I'm happy to talk more about this in the questions if you guys want, but we basically found iron supports DOP consumption in some circumstances, but when iron is really limiting, then basically it allows DOP to accumulate because there's not enough iron around to support DOP consumption, okay? So this is a project that I've been working on with my colleague and co-PI, Rob Letcher, who's at the University of New Hampshire. So he does modeling of dissolved organic matter stoichiometry. And what we've been doing with Rob, and this is a paper that came out last year, is working to quantify the role of dissolved organic phosphorus based on this data set and the work that we've all been doing in supporting rates of annual net community production, so rates of photosynthesis in the global ocean. And so here, Rob was modeling the DOP production flux. And in this panel, what you see is the autotrophic DOP uptake flux. So the rate of DOP consumption by phytoplankton. And there's other ways to visualize this. So here he's got model-based estimates of annual rates of net community production in phosphorus units, in carbon units. And here is the fraction of DOP-supported annual net community production. So that is to say, the fractional importance of DOP for supporting net community production in these regions on an annual basis. And so what you can see is that in the low latitudes and in the gyres, and especially in the Sargasso Sea, and especially as you get to the western side of the Pacific, even where we found these high rates of nitrogen fixation, DOP becomes the dominant source of phosphorus supporting carbon fixation, okay? And then the last thing that I want to talk about really quickly, and this is a paper that came out yesterday, and I'm so excited about this paper. So this is also work that my grad student has been leading and that Rob Letcher and I have been supporting, is we, again, are using this dissolved organic phosphorus concentration database to take a global look at surface ocean DOC to DON to DOP stoichiometry. And the idea here is that we want to compare carbon to nitrogen to phosphorus stoichiometry and dissolved organic matter with Redfield stoichiometry and spatially to try and get a sense of where certain things are, where dissolved organic nitrogen and phosphorus may be important for supporting phytoplankton growth. And so, you know, what this looks like is that you will have increasing DOC to DOP or DON to DOP concentration ratios as DOP is consumed. What we found is that DOP concentrations change much more than DOC or DON in the global surface ocean. And we think that that's because, the chemical composition of DOP allows it to be used as an assimilative phosphorus source much more easily than DON. DOC is just used by heterotrophs, not by autotrophs. And so what we found are a couple, I'm just going to give you the big punchlines here. And so we found zonal trends in the Pacific and meridional trends in the Atlantic. So here I'm showing you results from the Pacific. So this is longitude on the x-axis. So this includes all the data from both the North and the South Pacific. The left-hand y-axes are DON to DOP and DOC to DOP concentration ratios in the red. And then the black line is P star. So there's high amounts of phosphate in the east, which is consistent with denitrification, and then low amounts of phosphate in the west. And then you've got the water column denitrification rates in the blue, and then dust deposition rates in the gray. And then the green line is this remote sensing metric of iron stress as experienced by phytoplankton called FESAT. So that's something that Toby Westbury and Mike Barenfeld and others have developed out at Oregon State. But the idea here is that basically as phosphate concentrations decrease from east to west across the Pacific, you see the DON to DOP and the DOC to DOP concentration ratios increasing significantly. And to us, this is a signal. We interpret this to mean that phytoplankton are consuming dissolved organic phosphorus as an assimilative phosphorus source. But this is the first time we've been able to take this sort of large scale view using this new DOP data set and to be able to see these kind of global patterns. We have never been able to see this before. So we're really excited about this. In the Atlantic, what we see is something different. So now the x-axis is latitude, and we're starting in the Southern Ocean here, or southern part of the Atlantic, and going to the North Atlantic here. The red is still DON to DOP and DOC to DOP concentration ratios. The green is the FESAT iron stress metric. So the higher this number is, the more iron stress phytoplankton are experiencing. It's kind of like FV over FM as detected by satellite. The gray lines are atmospheric dust deposition, and then the black line is the excess phosphate metric, okay, P star. And what you can see is that there are higher DOC to DOP and DON to DOP concentration ratios in the north compared to the south. And especially in the Sargasso Sea, which is this region highlighted in light blue. And you see really high rates of atmospheric dust deposition to the North Atlantic here. So what we interpret the higher DOC to DOP and DON to DOP concentration ratios to result from is the alleviation of iron stress in the North Atlantic from atmospheric dust deposition. And this allows phytoplankton to consume DOP down to the lowest concentrations we see in the global ocean. And this is consistent with really decades of work done by people like Ben Van Mooy and Sonia Dyerman and Jill Sohm and Mike Lomas and others at the Bermuda site, and where they've done really careful studies and experiments with phytoplankton in at sea and in mesocosms and manipulative experiments on deck, where they've seen lots of evidence for DOP consumption by phytoplankton and diazotrophs in particular. Okay, so that's a lot about DOP. I do have a true love of dissolved organic nitrogen that I'm not really going to get to talk to you about, but one of the other things that came out of the work I did in the Eastern Tropical South Pacific is for the first time, we found evidence for consumption of DON with an isotope effect. So this really gives us a geochemical tool with which to track DON production and consumption. And we saw evidence for the first time in the Eastern Tropical South Pacific. And so now we're taking this and applying it to other ocean basins as well. And so this gives us a tool to see when DON is consumed. So I've talked to you a lot about our evidence for DOP consumption, and now we're using this metric for DON consumption, this isotopic change in DON consumption to help us understand when DON is being used. So that's something that I'm working on a lot going forward, is applying this tool elsewhere. Okay. And so with that, I will stop because I think I've at least talked for 30 minutes. So, and I will be happy to take questions from people.